all eukaryotes have mitochondria. So this should be a true statement. ये सही है या गलत है? And if I say कि some prokaryotes have mitochondria, some prokaryotes have mitochondria. ठीक है? And if I say कि some eukaryotes do not have mitochondria, so we can have certain kind of statements, So what do you think is most appropriate over here? We know all eukaryotes have mitochondria. This should be the right statement over here, but this is not right. This is a wrong statement, We know the exceptions in the eukaryotes. In fact, this should be the right option. Some eukaryotes do not have mitochondria. Okay, this is the right option. This is also wrong. This is the right option. We know all the eukaryotes, they do not have mitochondria. We have certain exceptions over here. We have certain exceptions. So, पहले तो हम यही क्लियर कर लेते हैं कि exceptions क्या हैं यहाँ पे. क्योंकि you go to books, you go to colleges, you go to schools, the teacher would tell you कि mitochondria is a eukaryotic organelle. All the eukaryotes have mitochondria. Yes, we have certain exceptions over here. We have certain protestas, We have certain protesta. For example, we have a protesta which is called as Giardia lamblia. We have a protesta which is called as Giardia lamblia. When I say protesta, we have something called as monera also. Certain students might be confused. What is monera? What is protesta? So monerans, basically prokaryotic cells, we call them as monera. If I say bacteria, if we have cyanobacteria, they're basically monera. When I say protesta, we know protesta is a single-celled eukaryotes. What is a protesta? This is a single-celled eukaryote. So what is the Giardia lamblia? It is a protesta. For example, I say Chlamydomonas. For example, I say Plasmodium. They're all, I say you have amoeba. It is a protesta. It is a single-celled cell. So similarly, we have Giardia lamblia. We know Giardia lamblia, it does not have mitochondria. It does not have mitochondria and is a eukaryotic cell. So this is one exception that we have. Okay. Now the question is, when I'm talking about, when I talk about Giardia lamblia, I'm saying it does not have mitochondria. There's no mitochondria. So if this organism does not have mitochondria, then how does it produce energy? How it produces energy then? How it produces ATP then? Tell me. क्योंकि mitochondria तो नहीं है इसमें. How does it produce ATP? We know mitochondria is the powerhouse of the cell. सब में रट्टा लगा है. We know mitochondria is the powerhouse of the cell. This is the one which produces energy. यहाँ तो mitochondria नहीं है. It is a eukaryotic cell. Then how the hell it produces ATP then? We know in this Giardia lamblia, we have certain special organelles. There are certain special organelles that we have, and we call these organelles as hydrogenosomes. What do we call these organelles? We have not heard of, many students have not heard of something called as hydrogenosomes. They're also the organelles, which are present in certain protista, certain fungi. They have hydrogenosomes. We know these hydrogenosomes can also produce ATP. They can also produce ATP, but along with ATP, they also liberate hydrogen. They also liberate hydrogen along with ATP. That is why we call them as hydrogenosomes. So even if they do not have mitochondria, they're supposed to have hydrogenosomes, which can also produce ATP. So this is one fact when we start with mitochondria. So now this is clear. All the eukaryotes, they do not have mitochondria. We have heard of nostoc. Okay. We have heard of certain bacteria. We know they do not have mitochondria. Then how the hell does the nostoc produce energy? And we know nostoc, for example, it is a BGA. We know it is a blue-green alga. When I say blue-green alga, matlab, it is also photosynthetic. It also photosynthesizes. It also produces energy. But we have no mitochondria. And we know it photosynthesizes. We know everybody has seen nostoc colony under the microscope. सबने nostoc देखा है. How does it look like? What is the color of nostoc? We know nostoc. ये देखो nostoc. Heterocyst, which fix nitrogen. We know heterocyst. Then again we have, what is the color? Don't you say green in color? Nostoc we know is green in color. But it is photosynthetic, we know. It is photosynthetic. Green. But you'll be amazed to know, you'll be surprised to know, there is no chloroplast. Then where are the pigments? If chloroplast nahi hai, then where are the pigments? Why does it look green? Mitochondria bhi nahi hai. Chloroplast bhi nahi hai. The bit is surviving. It is green in color. So what is going on? So you have to find it out. We'll discuss this nostoc in detail later. Okay? So I'm trying to tell you something, it's not necessary that we need mitochondria to survive. We know there are organisms, even if they have mitochondria. I say this is a eukaryotic cell. It has mitochondria. Okay, it has mitochondria. This is mitochondria. तो क्या? Do you think only mitochondria produces ATP? क्या सिर्फ mitochondria produce करता है? ऐसा तो नहीं है. It's not necessary that only mitochondria produces ATP. If I put this cell under anaerobic condition, if I put this cell under anaerobic condition, under anoxic condition, under anaerobic condition, we know there's no oxygen. And mitochondria requires oxygen to function. Mitochondria needs oxygen. Respiration होती है, oxygen चाहिए. Mitochondria needs oxygen. जब oxygen नहीं है, there's no oxygen, the mitochondria stops functioning. So do you think the cell will not produce ATP or what? Cell still produces ATP. This cell still produces ATP. How the hell it produces ATP? We have something called as glycolysis. Everybody has heard of glycolysis. Glycolysis. सबने पढ़ा है glycolysis. What is the location of glycolysis? Glycolysis is not found in mitochondria, my friend. Glycolysis is found in cytoplasm. Glycolysis is found in cytoplasm. And we know glycolysis, this also produces ATP. So, याद है कि नहीं है? We know glycolysis produces ATP. Glycolysis produces NADH. We know this. NADH is also energy rich. ATP is also energy rich. Then what? If mitochondria is not functional, it does not have any harm. The cell can still produce ATP. Through something, through a mechanism, it produces ATP. Through what mechanism? Through a mechanism which is called as substrate level phosphorylation. What do we call this? We call it substrate level phosphorylation. ATP can be produced. And mitochondria produces ATP. Okay. So, see, I'm trying to tell you, it's not necessary. ऐसा नहीं कि mitochondria is the only powerhouse of the cell it produces. If mitochondria will not be there, my cell cannot function. It's nothing so. We know in many tumorous cells, many tumors. In many tumors, we know they initially grow under anoxic conditions. Anoxic conditions, they are not very well supplied with the blood vessels. We know many tumors, they are not very well supplied with the blood vessels. They grow under anaerobic conditions. Anaerobic conditions. So what do you think? They'll die or what? They don't die. They can still survive. They still produce ATP. Through glycolysis, we know they still produce ATP through glycolysis. Look at this, they don't die. So this should be very, very clear. Okay. Moving forward, when I'm talking about ATP production, because if I ask you, what is the function of mitochondria? We know 95% students will say कि सर माइटोकॉन्ड्रिया का फंक्शन है एटीपी का प्रोडक्शन. यही बोलोगे आप कि mitochondria's function is to produce ATP. But do you think this is the only function of mitochondria? No. Mitochondria has more than 10 functions. We can have plethora of functions. We can have multiple functions of mitochondria. It is a multifunctional organelle. It is a multifunctional organelle. ऐसा नहीं है कि its only function is to produce ATP. Look at the ATP production. We know there are 3 methods of ATP production. There are 3 ways by which a cell can produce ATP. The first one you have just seen. The first method you have just seen. What is that? Substrate level phosphorylation. पहला method क्या है? Substrate level. मतलब in the first method, the ATP can be produced in cytoplasm. And what do we call it? We call it substrate level phosphorylation. We call it substrate level phosphorylation. The second method we know involves ATP production in mitochondria. This is also method. Mitochondria is involved. Mitochondria is involved over here. And what do we call this method is? The ATP produced by mitochondria. What do we call it? We call it oxidative phosphorylation. We call it oxidative phosphorylation. Oxidative phosphorylation. We also have a third method. Color से ही पता लग रहा है. Green. That means third method of ATP production in the chloroplast. If I ask anybody, which organelle produce mitochondria? You would say, sir, mitochondria. But we also have chloroplast. We know chloroplast can also produce ATP. क्या chloroplast में production नहीं होता ATP का? Chloroplast we know can also produce ATP होता है, लेकिन बच्चे आंसर नहीं देते. बच्चे सिर्फ माइटोकॉन्ड्रिया ही आंसर देते हैं. But we know chloroplast can also produce ATP. And what do we call this method? क्या कहेंगे? Photophosphorylation. All this as we call it photophosphorylation. Photophosphorylation. Look at different methods, you know. Substrate level, oxidative, photophosphorylation. We have different mechanisms that ATP production can take place here. Okay. तो चलो ठीक है. Because we are talking about mitochondria today, हम अभी बाकियों की बात नहीं कर रहे हैं. We are focusing on mitochondria. So when we talk about mitochondria, we know this organelle mitochondria, this was first observed. थोड़ी सी history देखते हैं. This was first observed by a scientist whose name is Kolliker. Kolliker. ठीक है. And ये जो name mitochondria, ये जो name mitochondria है, this name mitochondria, this was first given by a scientist called as Benda. He's the one who gave this name mitochondria, you know, Benda. And we know, everybody knows, ये जो mitochondria है, we use a stain. Which stain is used to analyze mitochondria? We know this is Janus green B vital stain. This is the stain which is used to stain mitochondria. Okay? So we know something about the mitochondria. We know something about the mitochondria. Okay? Now the question is, see, there's some puzzling. There's something which is puzzling me, you know. Something which is confusing me, you know. Something which confuses me. And what confuses me, we know, if there's a human or if there's an animal, you treat him with cyanide. You ask him to eat cyanide, you know. Do you think he can tell the taste of cyanide? If you feed an animal, if you feed humans with cyanide, do you think they can tell you the taste of cyanide?
No, sir.
No.
No, sir.
They will die. We know they will die. What does the cyanide do? Cyanide is a respiratory poison. We know cyanide is a respiratory poison. What is the target site of cyanide? Cyanide कहाँ जाके क्या करता है cyanide? We know the target of cyanide is basically mitochondria. Target of cyanide is mitochondria. It is mitochondria. तो mitochondria में क्या करेगा? Where in the mitochondria is the site of cyanide action? अगर मैं mitochondria की बात करूं तो mitochondria में कहां पे जाके inhibit करता है? What is the site of action of cyanide? We know it is the electron transport chain. It is the ETC. Look at the ETC. हमको सबको पता है ये outer mitochondrial membrane है, ये inner mitochondrial membrane है, ये sorry, this is the outer mitochondrial membrane, this is the inner mitochondrial membrane, this is the matrix that we have. Everybody knows the inner mitochondrial membrane has electron transport chain. सबने देखा है. We have the ETC. ETC. Is it right or wrong? What I've drawn is right or wrong? Tell me.
So shouldn't it be present in the intermembrane space?
No, this is internal membrane only. They're all present in the internal membrane, and most of them are integral membrane proteins. That means they'll protrude from both the sides.
Sir, 4 complex only, sir. This third one is inside that
Yeah, very good.
CO2 is inside.
Yes, very good. That means we only have 4 complexes. We don't have 5. This is the answer. I have drawn 5. In certain books, they mention 5. You go to some Indian authors, they have mentioned, okay, mitochondrial ETC is made up of 5 different complexes. No, that is wrong. We know mitochondrial ETC only has 4 carrier complexes. This is 1, this is 2, this is 3, this is 4. There's nothing called as 5. This is ATP synthase. Some people call it 5. It's not 5. It is simply what is F0F1 particle, which is ATP synthase. Okay? So look at this. When I'm talking about this, ultimately what I'm saying is कि site of the action of cyanide, ये हमारा cyanide है. This is cyanide that we have. ठीक है? This is the cyanide. So where does the cyanide act? The site of action of cyanide is the carrier complex 4. यहाँ पे आके इसको inhibit करता है. It basically inhibits carrier complex 4. So ETC will not take place. ATP cannot be synthesized. So this is very true. But the question is, why it is confusing? Why it is confusing me? We also have plants. Plants also have mitochondria. They have the same ETC. Same electron transport chain is present here also. But the problem is, when you treat plants with cyanide, when I treat this with cyanide, plants don't die. Do you think plants will die? Do you think plants will die if you treat them with cyanide?
No, sir.
क्यों? क्यों? Why? Same ETC is here also. Same ETC here also we have. ठीक है? The question is, why don't the plants die? So this is something which is puzzling me. Plants normally don't die because of cyanide poisoning, because of azide poisoning, because of carbon monoxide poisoning. You know, all these chemicals can target mitochondria, whether it is cyanide, whether it is carbon monoxide, whether it is azide. They all inhibit mitochondrial ETC. But why not plants? That means when we study about mitochondria, there must be certain differences between plants and animals. There must be certain differences between animals and plants. And this is why what we don't study in colleges. हम वही पढ़ते हैं, चाहे वो बॉटनी वाले हों, चाहे zoology वाले हों, चाहे biochemistry वाले हों. They study same books, they study same everything. But we know there are significant differences between plant and animal mitochondria also. Okay. So see, when we start with mitochondria, the question is, when we start with mitochondria, we know there are various functions of mitochondria. There are various functions of mitochondria that we normally don't study in colleges. ठीक है, एक बारी IAS के एग्जाम में they have asked कि okay, discuss about different functions of mitochondria. There was a question of 20 marks in IAS examination, discuss different functions of mitochondria. And what the students have done, उन्होंने क्या किया कि पूरा mitochondria explain कर दिया, पूरा glycolysis, पूरा electron transport chain, पूरा what we have TCA cycle, they have discussed. Who asked you? The examiner simply asked functions of mitochondria, and you discussing all this glycolysis and its regulation, all nobody asked you. So you have to look at the question first. So when I say different functions of mitochondria, we know, okay, the first function, as you all know, this is ATP production. And any more functions of mitochondria that we have? We know mitochondria, as you all know, is also involved in what? We know it is involved in fatty acid oxidation. Fatty acid oxidation. That means breakdown of fats. Fat breakdown, fatty acid oxidation. This is also one of the function of mitochondria. And what do we call it? We call it beta oxidation. We call it beta oxidation. This is also a function of mitochondria. Beta oxidation होती है, fatty acid का breakdown होता है mitochondria में. And when the fatty acids are broken down, we get 2 carbon subunits. This will liberate 2 carbon subunits. We call them as acetyl CoA. We get acetyl CoA, which are the 2 carbon compounds. So this is also a function of mitochondria. Now, if I ask you in plants, when the plants, the fatty acid oxidation will take place, tell me. Bolo. Plants. The answer should be same. In plants also, we have mitochondria. This is half the answer. Mitochondria is right, but we know this is half the answer. In plants, we know beta oxidation can also take place in peroxisomes. Peroxisomes or glyoxysomes. Glyoxysomes. The plants have 2 organelles where beta oxidation can take place. Some other functions of mitochondria. We also have amino acid oxidation. Mitochondria is also involved in amino acid oxidation. Mitochondria is also involved in fatty acid chain elongation. Fatty acid chain elongation. Mitochondria is also involved in the breakdown of tryptophan. Mitochondria is also involved in the oxidation of epinephrine. Mitochondria is also involved in certain anaplerotic reactions. मतलब synthetic reactions. Synthetic reactions. ठीक है? We'll be talking about them later. So look at different functions of mitochondria. Another function of mitochondria, we know mitochondria, very important. Mitochondria is also involved in glyoxylate cycle. Partly involved in glyoxylate cycle, especially in the case of plants. Another function of mitochondria. Can you name some functions? Can you tell me some functions of mitochondria? Mitochondria is also involved in photorespiration. Photorespiration. Again in plants. You'll say, sir, what is photorespiration? Can you tell me what is photorespiration? स्पेशली रिमेंबर 12th क्लास में आपने रट्टा लगाया था कि फोटो रेस्पिरेशन इज अ नेसेसरी इविल और समथिंग लाइक दैट. Especially it takes place in the C3 plants. Photo respiration. More prominent. It is more prominent. So let's not discuss in detail, but just remember, they'll also participate in photo respiration. Another function of mitochondria. We know mitochondria has a very important role. They also have a role in cell death. That is apoptosis. Apoptosis. Mitochondria, जो membranes हैं, mitochondria membranes have special proteins which are called as UCPs. UCPs, mitochondrial membranes, have proteins, especially in mammals, small mammals. They have proteins called as UCPs, uncoupling proteins. What is UCPs? They are the uncoupling proteins. And we know these uncoupling proteins, they are involved in production of heat. Production of heat. And that is how they regulate the body temperature in small mammals. They regulate body temperature. Small mammals. Look at various functions, my friend. और भी बहुत सारे functions हैं. 12th. We know what we have seen. कभी अगर मैं कहूंगा you'll see, सर ये क्या बोल रहे हैं आप? You know, your body can increase temperature. हमें पता है आपको फीवर होता है, आपकी बॉडी का टेम्परेचर इंक्रीज हो जाता है. 37 से 39 हो गया, 98 for night से it reaches 102, 103 high temperature. But if I say कि plants can also increase temperature, ये प्लांट भी गर्म हो गया, आप कहोगे सर ये क्या मजाक कर रहे हो आप? Yes, we know there are certain plants which can also increase their temperature. They can also increase their temperature. And we call this phenomena as thermogenesis. We call this phenomenon as thermogenesis. I'll give you an example. One example is arum lily. We know that during time of reproduction, during time of pollination, the temperature of the plant increases. The temperature of the plant increases. We call it thermogenesis. Who is involved? Who is involved in thermogenesis? We know it is mitochondria. Mitochondria. देख रहे हो आप? So do you think only ATP production is the function of mitochondria? नहीं. Mitochondria has various functions. Am I clear with this? और भी कई सारे functions हैं. जैसे जैसे we'll see mitochondria, you'll see more functions of mitochondria. You can analyze yourself. So when we are talking about mitochondria, the question is, अब हमें पता लग गया mitochondria important है, बहुत सारे functions हैं mitochondria के, ठीक है? लेकिन when we come down to the structure of mitochondria, structure of mitochondria, nothing new. Everybody knows the structure. It is a double membrane organelle. We know mitochondria is a double membrane organelle. ठीक है? तो भाई ये outer membrane है हमारी. We know all this. We have the outer membrane. We have the inner membrane, which is folded. We know inner membrane is folded into structures which are called as cristae. Cristae. Foldings are called as cristae. And this is the intermembrane space we have. This is the intermembrane space. And this is acidic. We know intermembrane space is acidic in nature. A lot of H-ions are present here. A lot of H-ions are present here. Now also attached to the inner membrane, we have small particles. Attached to the inner membrane, we have small particles. ये भी आपको पता है. This is F0, F1 particle. F1 particles. We call them as ATP synthesis. ATP synthesis. Or sometimes we have another name for them. Sometimes we call them as Fernandez-Morans particles. Fernandez-Morans particles. We have another name. We can call them as oxysomes. Oxysomes. तो ये एक ही है सब ये this is not your level. This is 12th class level. ये medical में you mug up all these things, but now you are at higher level. You have to go for more analysis now. Okay. So we know F01 particles, and inner membrane encloses a fluid-like, substance which is matrix. So we have matrix over here. Is this clear? And by the way, mitochondria we know is semi-autonomous. Everybody knows this is a semi-autonomous organelle. Semi-autonomous organelle. That means it has genome of its own. इसका खुद का भी genome है. We know it is DNA. Now what kind of DNA? DNA, is it circular? Is it linear? What kind of DNA do we have? We have circular DNA. What kind of DNA do we have? We have circular DNA. How many copies? Do you think only one copy? No, we have multiple copies. We have multiple copies of circular DNA. So look at this. This is also part of the structure. Now the question is, does mitochondria have ribosomes? Does mitochondria have ribosomes? Yes. We know mitochondria, in fact, it evolved from a prokaryotic cell. We know mitochondria basically evolved from a prokaryotic cell. We have something called as endosymbiotic theory. We have something called as endosymbiotic theory. So ultimately, because prokaryotes have ribosomes, and because mitochondria has evolved from a prokaryotic cell, that means mitochondria is also supposed to have ribosomes. That means mitochondria has ribosomes. We have ribosomes also. So what kind of ribosomes are they? 70S or 80S? What kind of ribosomes do we have?
70.
Somebody says this is wrong, so you'll say 80S. That is also wrong. Lekin prokaryotes have 70S. Prokaryotes have 70S. Then what kind of ribosomes does mitochondria have? Now let me tell you, ये जो mitochondria है, they have ribosomes which are 55S. Keep this in mind. 55S ribosomes are present over here. Large subunit, small subunit. We have a large subunit which is 39S, and we have a small subunit which is 28S. So total this is 55S that we have. This is the mitochondria, ribosome. Am I clear? Okay. So we have seen.
Excuse me, sir.
Yeah.
Sir, how many large subunits are there? Is it 28 or 39?
How many?
The large subunit, was that
This
39
is
or
39. 39. 28 is small.
okay.
39 is larger.
Okay, sir. Right.
Okay. So this is very clear because ये question आया हुआ है, and most of these students would go for 70S, but that is the wrong answer. The answer is what is 55S. Okay. So we have seen the basic structure of what is the mitochondria. Now the question is, when you look at the membranes of mitochondria, so nothing much to tell. When I look at the membranes, you all know, you're all smart. We have two membranes. We know it is a double membrane organelle, like chloroplast is also a double membrane. We have mitochondria is also a double membrane organelle. There are two membranes that we have. This is the outer membrane. This is the inner membrane. We all know the outer membrane. Now, there should be clarity over here. The outer membrane is also bilayered. अगर ये membrane है, तो this is bilayered. मतलब this membrane is a bilayer. ऐसा नहीं कि this is a bilayer. Similarly, inner membrane is also a bilayer. So that means both the membranes are in fact bilayers. Both these membranes are bilayers over here. Outer membrane is bilayer, inner membrane is bilayer. But if you look at the membranes, if you look at the composition, if you look at their protein lipid ratio, we see there are starking differences, there are differences between the two membranes. We'll see there are many differences between the two membranes. If you look at the outer membrane, if I look at the outer membrane, we know outer membrane is approximately 50% lipid, by weight. This is approximately 50% lipid by weight. Not only this, we know ये जो outer membrane है, this outer membrane also contains various enzymes. The outer membrane, ये हमारी जो outer membrane है, this outer membrane also contains various enzymes. Which enzymes? For example, the outer membranes has enzymes which are involved in oxidation of epinephrine. It has enzymes involved in oxidation of epinephrine. It has enzymes which are involved in elongation of fatty acids. It has enzymes which are involved in the breakdown of tryptophan. Breakdown of tryptophan. Look at this. Outer membrane. And at the same time, ये जो outer membrane है, if you look at the outer membrane, we know outer membrane has transport proteins. It has transport proteins which are called as porins. Outer membrane has transport proteins which are called as porins. ये जो porins हैं, they're absent from the inner membrane. Absent from the inner membrane. Look at the differences. Porins are present in the outer membrane. In fact, ये जो porin proteins होते हैं, they are made up of not alpha helical, but they are beta pleated sheet structure. याद रखो ये जो porins होते हैं, these porins are made up of beta pleated sheet structure. Yeah, biochemistry में you'll study all this in detail. They are made up of closed beta pleated sheet structure which are closed. ये closed हैं, देखो गोले हैं, circular. That means closed. Sheet-like structure has been closed to form a circular structure. And we call them as what? We call them as beta barrels. We call them as beta barrels. So that means ये जो porins हैं, in fact, they are beta barrels. They're beta barrels. Is this clear? And for many years, what we used to think, ये जो porins हैं, they were found to be always open. ठीक है? These porins are found in bacteria also. Porins are found in our plasma membrane also. But if you look at these porins in the plasma membrane, we know they're always open. They're always open. But porins which are present in mitochondria, we know they can open and close. They can undergo reversible closure. They can undergo reversible closure in response to conditions within the mitochondria, in response to conditions within mitochondria. So they can open and close. They can open, they can close. This is in contrast to the other porins. Okay. So we're looking at the outer membrane. That means because these porins are present over here, the outer membrane comparatively is permeable. Solutes can enter. Solutes can enter. It is comparatively more permeable in comparison to the inner membrane. It is found to be more permeable because of the presence of these transport proteins called as porins. And by the way, ये जो porins हैं, these porins are nothing else. The porins are channel proteins. They are channels. They are examples of channel proteins. Under channel proteins, we also have ion channels. So ion channels, porins, all are under channel proteins. Let's not confuse. We just know we're looking at the membranes. Let us keep it simple. Let us not make it complex. So now we know the outer membrane, more rich in lipids, more permeable, presence of porins, certain functions, different enzymes are associated with the outer membrane. Enzymes involve oxidation of epinephrine, elongation of fatty acids, breakdown of tryptophan. Let us look at the inner membrane now. Let us look at the inner membrane. Inner membrane of mitochondria. When we are talking about the inner membrane of mitochondria, the first thing you have to remember, we know, look at the inner membrane. The inner membrane has the entire electron transport chain. F0, F1 particle. So that means ये जो inner membrane है, it is rich in proteins. It is rich in proteins because of the presence of different components of ETC. Different components of ETC, it is rich in proteins. And this is what we have seen, कि inner membrane में, if you look at the inner membrane, we have more than 100 different polypeptides, more than 100 different polypeptides, which are found in the inner membrane. And if you look at the protein lipid ratio, अगर मैं protein lipid ratio की बात करूं inner membrane में, we know protein lipid ratio is very high. It is approximately 3:1. मतलब we have 3 times more proteins in comparison to lipids in the inner membrane. Look at this. So protein lipid ratio is approximately 3:1. This is by weight, not by numbers, by weight. Okay. So look at this. That means inner membrane has more proteins. Certain more differences. What we have seen, ये जो inner membrane है, now this is very important. Inner membrane is completely devoid of cholesterol. There's no cholesterol in the inner membrane. We don't have any cholesterol. Whereas the outer membrane is found to have cholesterol. Outer membrane of mitochondria is found to have cholesterol. Inner membrane has no cholesterol. In fact, a normal plasma membrane, अगर मैं plasma membrane की भी बात करूं, मैंने कहा हमारी plasma membrane है, ये हमारी cell की plasma membrane है, cell membrane, plasma membrane, whatever you call it. ठीक है? So plasma membrane we all know has lipids. It has proteins, it has carbohydrates. सबको पता है, lipids भी होते हैं, proteins भी होते हैं, carbohydrates भी होते हैं plasma membrane में. Lipids में भी we have, see, look at the lipids. We have phospholipids. सबको पता है plasma membrane में phospholipids होते हैं. We have glycolipids also. And we also have sterols. मतलब cholesterol. तो plasma membrane, normal membrane has cholesterol. This is a normal feature. ऐसा नहीं कि cholesterol आपके blood में ही होता है सिर्फ, मम्मी के blood में, दादाजी के blood में cholesterol बढ़ गया. No, cholesterol is an integral part of membrane. Both the monolates can have cholesterol. Cholesterol यहाँ पे भी हो सकता है, यहाँ पे भी हो सकता है. दोनों जगह cholesterol हो सकता है. So cholesterol we know is an integral part of membrane. So it is a normal feature. To have cholesterol is normal. But look at the mitochondrial membrane. The inner mitochondrial membrane, I am saying, has no cholesterol. This is something which is abnormal. This is not normal. This is something which is exception. This is something which is exceptional. It has no cholesterol, whereas the outer one has. Look at one more difference. We know inner membrane is found to have a special phospholipid. अभी हमने देखो phospholipid की बात करी थी. I told you phospholipids. I told you membranes have phospholipids. Plasma membrane has phospholipid. Now we inner membrane of mitochondria. I hope I am not fast. किसी को लग तो नहीं रहा कि मैं ज्यादा तेज मूव कर रहा हूं? You can interrupt me. अगर किसी को हिंदी अंग्रेजी समझ में नहीं आ रही, you can ask me. I can speak in Hindi also. The main thing is you have to understand. That is more important. Language is never a barrier. If Rajnikant speaks Hindi, everybody understands. South Indians or North Indians, everybody can understand. So I think so. Language is not a barrier. Some people can always understand little of English, little of Hindi. Everybody can understand. Okay, Punjabi, we can understand a little bit of Punjabi also. Now the question is, when we are talking about inner membrane, let us see this. When I'm talking about inner membrane, the inner membrane is found to have a special phospholipid. It is found to have a special phospholipid. And what is that? That is called as cardiolipin. Cardiolipin. This is absent in outer membrane, absent in plasma membrane. Plasma membrane में भी नहीं है. It is found in the inner membrane of mitochondria, a cardiolipin. The scientific name of cardiolipin is, this is diphosphatidylglycerol glycerol. Scientifically हम क्या कहते हैं इसको? Scientifically we call it diphosphatidylglycerol. Sorry, मैं cholesterol लिख दिया. This is diphosphatidylglycerol. Glycerol. This is what is cardiolipin, found in the inner mitochondrial membrane. And we know inner mitochondrial membrane also has ATP synthase. It also has F01 particle. What scientists believe? What scientists believe that the function of this cardiolipin, what they say, what is the function of this? What is the importance of this cardiolipin? The function of this cardiolipin is to assist, is to facilitate, is to help those proteins which are involved in ATP synthesis. It is known to facilitate the activity, it is known to help in the activity of those proteins which are involved in ATP synthesis. Am I clear? So look at the differences between outer and inner membrane. Not only this, ये जो inner membrane of mitochondria है, inner membrane of mitochondria is highly impermeable. Highly impermeable. Impermeable. ठीक है? So we have seen certain differences between the outer and the inner membrane. Is this clear? So we're starting with the mitochondria. We have the DNA. We know multiple copies of circular DNA. We have the F01 particle. We look at the structure of the F01 particle later. Now, the question is, when we talk about mitochondria, what we know, कि mitochondria में, we know one of the main functions is ATP production. We know one of the main function is ATP production. And when we talk about ATP production, the ATP production itself can be divided into 5 processes. There are 5 processes which are involved. There are 5 reactions which are involved, different set of reactions which will lead to production of ATP. We sometimes call it mitochondrial respiration. When I say mitochondrial respiration, मतलब क्या? मतलब production of ATP, breakdown, production of ATP. And we know there are 5 set of reactions which are involved. In mitochondrial respiration, we have 5 different set of reactions. पहला क्या है? We all start with glycolysis. We know we all start with glycolysis. Glycolysis. कैसे start करते हैं? यहीं से start करेंगे glycolysis से? How you start mitochondrial respiration? Tell me. This is the first set of reaction that we study, glycolysis. And we know ये जो glycolysis है, this glycolysis, it is located, it is found in cytoplasm. Cytoplasm. It's not found in mitochondria. So, but this is half the answer. When I say glycolysis is found in cytoplasm, this is half the answer. So what is the full answer? This is half the answer. The question is, I told you, differences between animals and plants. When I talk about plants, if you see glycolysis in plants has dual location, 2 locations. One is cytosol, and the second is plastids. Plastids. So plants में we have dual location of glycolysis. One is cytosol, one is what is plastids. This should be very, very clear. Okay. So when we talk about glycolysis, हम जब glycolysis की बात करते हैं, we know taking place in cytosol normally. And how does the glycolysis start? We all know it starts with glucose. Starts with glucose. आपने खाना खाया, खाने से ग्लूकोज आया, ग्लूकोज ब्लड में आया, your tissues, your cells will get glucose. Sometimes you're starving. मम्मी ने आज खाना नहीं बनाया, मम्मी की पापा से लड़ाई हो गई, or you're doing long fasting, or you're doing rigorous exercise. So in those conditions, what will happen? ये जो blood में glucose है, that blood glucose would be depleted. But there are certain tissues which need glucose. Blood में तो glucose है नहीं, and that is how your body breaks down stored glycogen. Your body breaks down stored glycogen. So that means it can start from here also. तो यहाँ से भी हम glycolysis start कर सकते हैं, यहाँ से भी start कर सकते हैं. We know glycogen when broken down will also give rise to glucose. Is this clear? So both ways. We can start with glucose also, we can start with glycogen also, but there are certain differences. We'll see this later. There are certain differences which are involved. The question is, when we are talking about glycolysis, if you look at the plants, how do we start in plants? Tell me. How do I start in plants? If you're a student of botany, or maybe you're a student of plant biotech, or maybe you never know, people of animals start working in plants when they do their PhD research. So how do we start with glycolysis? Shall I start with glucose? Tell me. This is nothing new. मैं कुछ ऐसा नहीं पूछ रहा हूँ जो आपको नहीं पता है. आपने तो ये पढ़ा है सब कुछ. 12th क्लास में भी रट्टा लगाया होगा, 10th क्लास में भी, BSc में भी पढ़ा होगा, MSc में भी बहुत लोगों ने पढ़ा हुआ है, जो कुछ लोग हैं. So how do we start glycolysis in plants? See, if you talk about plants, we have photosynthesis. Plants में photosynthesis होती है. तो photosynthesis का product क्या है? What is the end product of photosynthesis? Do you think this is glucose? Do you think end product of photosynthesis is glucose? Tell me, what is the end product of photosynthesis?
Glucose plus oxygen.
क्या? बोलो बोलो फिर से बोलो.
Sir, glucose plus oxygen.
Sucrose or starch.
I'm talking about oxygen is, we know, it is the end product of photosynthesis, but we are not concerned with the oxygen. We are concerned with this energy rich molecule. This is not glucose. In fact, let me tell you, you'll be surprised to know this, that the end product of photosynthesis is never a hexose sugar. It's a triose phosphate. The end product of photosynthesis, in fact, is a triose phosphate, 3-carbon compound. And later, जो 3-carbon compound है, this is either converted to starch, or this is converted to sucrose, or this is converted to, in certain plants, small number of plants, we also have compounds which are called as fructans. Look at this. These are the end products, of photosynthesis. जो photosynthesis से खाना मिलता है हमें, you get the food in what form? Either this is starch, or this is sucrose, or these are fructans. So how do we start the glycolysis in plants? यहीं से start करेंगे, क्योंकि this is the stored form. So when I start glycolysis in plants, I have to either start with, yes, my friend, sucrose. Sucrose is very important. Very, very important. Always gets, we know it is a disaccharide. This is a disaccharide. It is made up of fructose plus glucose. Sucrose is a disaccharide. It is made up of fructose plus glucose. अब देखो ये प्लांट है हमारा. ये हमारा प्लांट है. ठीक है, ये वाला बनाते हैं यार. ये तो हमने ट्री बना दिया, चलो ऐसे लेते हैं. दिस इज अ प्लांट, these are the leaves that we have. ये प्लांट है हमारा, ये लीव्स हैं यहाँ पे. ठीक है, ये हमारी रूट्स हैं यहाँ पे. These are the roots that we have. ठीक है, ये हमारी स्टेम है, यहाँ पे these are the roots. अब photosynthesis everybody knows takes place in the leaves. यहाँ पे photosynthesis हो रहा है. But root से I am also hungry. मुझे भी खाना चाहिए. Kitchen कहाँ है? Kitchen is in the leaves. Photosynthesis takes place in leaves. It is the kitchen of the house. Now the question is, kitchen में खाना बना. The khana has to be sent to roots also. The food which is synthesized in the leaves has to be sent to different parts of the plant. So what is that food? That is sucrose. Sucrose. Matlab, sucrose, always remember, is the transportable form of the sugar. What is this? This is transportable form of sugar. This is the transportable form of sugar. If food has to be transported from one part of the plant to another part of the plant, I say from leaves to the roots, in what form it will be transported? It will be transported in the form of sucrose. Sucrose. That means if I start glycolysis in the roots, अगर मैं roots में glycolysis start करूं, तो roots में जो glycolysis है वो कहां से start होगा? Start from sucrose. यहीं से start होगा. Am I clear? And when photosynthesis is taking place, not everything is sent to the plant. If during photosynthesis a plant synthesizes more food, अगर ज्यादा खाना बन गया तो क्या मम्मी फेंक देती हैं? आज ज्यादा बेटा चावल ज्यादा बन गए, सब्जी बन गई तो फेंक देते हैं, चलो. ऐसे तो नहीं होता. Your mother will store it in the refrigerator or something, or she'll give it to somebody, she'll store it. That means some amount of sugar, some amount of food that is being synthesized in the leaves, it is also stored in the plastids. Plastids के अंदर ही स्टोर कर लिया हमने क्लोरोप्लास्ट के अंदर ही स्टोर करा इसको. किस फॉर्म में? इन द फॉर्म ऑफ स्टार्च. So very important. What I'm trying to tell you, two molecules. Starch. What is starch? Starch, do you think it is a transportable form of sugar? No. This is storage form of sugar. This is storage form of sugar. And what is sucrose? This is the transportable form of sugar. Transportable form of sugar. This difference you have to keep in mind. And where is synthesis? ये question आता है बार-बार. Starch का synthesis कहाँ होता है? Where does starch synthesis takes place? Inside the leaf. This is in the plastid. मतलब inside the plastid, inside the chloroplast. And where is the sucrose synthesis taking place? Sucrose synthesis will take place in the cytosol. Keep this in mind. Cytosol. So when I start with glycolysis in the plants, we'll never start with glucose. कोई भी अगर बच्चा जो plants
of
of
chloroplast
क्या?
only.
अरे क्लोरोप्लास्ट में कब से साइटोसॉल आ गया? क्लोरोप्लास्ट में तो स्ट्रोमा होता है.
Yes, sir.
ये सेल है, ये क्लोरोप्लास्ट है हमारा. ये देखो हमारा क्लोरोप्लास्ट है ना, ग्रेना स्ट्रोमा. ठीक है, यहाँ पे ये ग्रेनल मैट्रिक्स, यहाँ पे स्ट्रोमा है हमारा. ये क्या है? ये हमारा न्यूक्लियस है. ये क्या है? ये साइटोप्लाज्म है. तो यहाँ पे है हमारा सुक्रोज. कहाँ पे है? Here. You got it now? ठीक है. तो एंड प्रोडक्ट क्या था फोटोसिंथेसिस का? एंड प्रोडक्ट इज ट्रायोस फॉस्फेट. मैंने क्या बताया था? व्हाट इज द एंड प्रोडक्ट ऑफ फोटोसिंथेसिस? ट्रायोस फॉस्फेट. तो फ्रॉम क्लोरोप्लास्ट ट्रायोस फॉस्फेट इज एक्सपोर्टेड टू साइटोप्लाज्म. It will come here and this triose phosphate is converted to sucrose. You get my point now? ठीक है. So this is very clear. So the question is, when I'm talking about glycolysis, to fetch you more marks, you can always draw some comparisons. कि plant में देखो ऐसा होता है, animals में ऐसा होता है, you'll get more marks now. So how do we start glycolysis in plants? Either we start from sucrose, or we start from starch, or we can start from fructans. यहां से start करना पड़ेगा, ग्लूकोज से नहीं करना पड़ेगा. And we know we have enzyme called as invertase. Invertase can break down sucrose into glucose plus fructose. And what is starch, by the way? Starch is a polymer of glucose. What is starch? Glucose, 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 glucose. It is a polymer of glucose. So when you break down starch, what do we get? My friend, what do we get? When I break down starch, I get glucose. Fructans are the polymers of fructose. So when I break down fructans, what do I get? I get fructose. Look at this. That is how we start the glycolysis. That is how we start the glycolysis over here. Very simple. Any confusion? So now we know. Animals, normally glucose. And we know this is glycolysis. We're talking about glycolysis. I'm not talking glycolysis in detail. I am just giving you a trailer today. मेरा काम पढ़ाना नहीं है, आज मैं आपको सिर्फ एक ट्रेलर दे रहा हूं यहाँ पे, क्योंकि माइटोकॉन्ड्रिया इज अ वेरी लॉन्ग टॉपिक. मैं तो आज को सिर्फ एक ट्रेलर दे रहा हूं कि क्या है, जस्ट टू मेक थिंग्स इंटरेस्टिंग फॉर यू. We know glycolysis is a 10-step reaction. This is a 10-step reaction. There are 10 steps which are involved in glycolysis. Glucose is a 6-carbon compound. So we know glucose is converted to glucose 6-phosphate. Glucose 6-phosphate is converted to fructose 6-phosphate. This is converted to fructose 1,6-biphosphate. So certainly we have, this is converted to 3-phosphoglyceraldehyde plus DHAP, dihydroxyacetone phosphate. 3-PGAL is again converted to, this is 1,3-bi-PGA, 1,3-biphosphoglyceric acid. This is converted to 3-phosphoglyceric acid. This is converted to 2-phosphoglyceric acid. This is converted to phosphoenol pyruvate. And finally, we get pyruvate. End product of glycolysis. Pyruvate is a 3-carbon compound. Glucose was a 6-carbon compound. And that is why we get 2 molecules of pyruvate. We start with 6 carbon, we end with 3 carbon. That is why we get 2 molecules. Rough idea, just a rough idea. Okay? And in between this, these reactions, we know the certain reactions which consume. So glycolysis में ऐसा नहीं है कि glycolysis only produces ATP. No. ये जो glycolysis है, we know glycolysis can consume ATP also. It can consume ATP also and can produce ATP also. तो दोनों हो रहे हैं यहाँ पे, कंजम्पशन भी है और प्रोडक्शन भी है. ये देखो जरा, ग्लूकोज टू ग्लूकोज 6-phosphate. ये फॉस्फेट कहां से आ गया? ATP converted to ADP. We get phosphate. Fructose 6-phosphate to fructose 1,6-biphosphate. एक एक्स्ट्रा फॉस्फेट आ गया यहाँ पे, देखो एक्स्ट्रा फॉस्फेट. हाउ डू वी गेट दिस? अगेन ATP is converted to ADP. So glycolysis is also consuming ATP. In the next set of reactions, it produces ATP. PP is a 3-carbon compound. ये सारे 3 carbon है. अब तक ऊपर सारे 6 हैं, ये 3 carbon है. So if you look at this 1,3-bi-PGA to 3-PGA, we know this step 1,3-bi-PGA to 3-PGA, ये वाला step है ना जो, we know this gives you 1 ATP. यहाँ पे, ठीक है, यहाँ पे, sorry, यहाँ पे PGAL, यहाँ पे, yeah, you get 1 ATP. Last step, look at phosphoenol pyruvate to pyruvate. यहाँ से phosphate हट गया, PEP2. Here also you get 1 ATP. यहाँ पे भी ATP बन रहा है, यहाँ पे भी ATP बन रहा है, दोनों जगह 2-2 ATP बन रहा है, यहाँ पे भी 2, यहाँ पे भी 2. तो टोटल प्रोडक्शन कितना है? 4 ATP का. टोटल कंजम्पशन कितना है? 2 ATP का. Consumption 2 ATP, production 4 ATP. So what is the net gain? Net gain we know is 2 ATP. We'll all see in detail later, don't worry. The question is, what I'm trying to tell you, कि जो glycolysis है, this glycolysis, it produces 4 ATP and it consumes 2 ATP. So that means there's a net gain of 2 ATP. And what is the end product? The end product is What is? देख रहे हो क्या है? Pyruvate. The end product is we also get 2 molecules of pyruvate. And we started with a molecule of glucose. And at the end, we get 2 molecules of pyruvate. All in cytoplasm. सब कहाँ हो रहा है? Cytosol. ATP मिल गया हमें cytosol में. We get the ATP in the cytosol. Look at the pyruvate. This is pyruvate. CH3, C double bond O, COO negative. This is what is a pyruvate, which is a 3-carbon compound. कहाँ पड़ा हुआ है? This is in cytosol. This is mitochondria. How do we move now? How do we move? So pyruvate, we know, is converted to acetyl CoA, which is a 2-carbon compound. And now this acetyl CoA enters the mitochondria. Right or wrong? Yes, all my new friends. Right or wrong? अरे कुछ तो बोलो. And now this acetyl CoA, when it enters mitochondria, it combines with oxaloacetic acid, which is a 4-carbon compound. This is a 2-carbon compound, and we get citrate. And that is how the TCA cycle begins. Citric acid cycle. Right or wrong?
Yes, sir.
Wrong. यहीं तो
Sir,
examiner यहीं तो
that is,
examiner this is
isn't that how it happens? So,
प्लीज बैकग्राउंड में किसकी आवाज आ रही है? अपना माइक प्लीज ऑफ कर लो आप. See, this is wrong, and this is where the examiner will try to confuse you. तभी मैं आपसे ऐसे, मैं आपको इसलिए बता रहा हूं, आई जस्ट वांट टू सी, और आप गलत आंसर दे रहे हो. What enters mitochondria is not acetyl CoA. What enters, yeah, see this. This is glycolysis. Glucose. We get 2 molecules of pyruvate. And at the same time, we also get net gain of 2 ATP, plus also we have a net gain of 2 NADH. This is the net gain. And now what happens? ये जो pyruvate है, pyruvate from cytoplasm is the one which enters the mitochondria. Always keep this in mind. It's not acetyl CoA which enters mitochondria. Question आ जाता है ये. What enters mitochondria? Not acetyl CoA. It is pyruvate, which is a 3-carbon compound. Am I clear now? And in the mitochondria, now we start with the second reaction. पहली reaction क्या थी हमारी? Glycolysis. This is the second reaction. What is the second reaction of respiration? The second reaction involves conversion of pyruvate into acetyl CoA. This is the second reaction. Conversion of pyruvate to acetyl CoA. Pyruvate, 3-carbon compound. Acetyl CoA, 2-carbon compound. Which is the enzyme? Enzyme is called as PDC. Pyruvate dehydrogenase complex. What is this PDC? ये नाम भी इतने बड़े हैं ना, ये लिखते लिखते आधा टाइम वेस्ट हो जाता है. Pyruvate dehydrogenase complex, PDC. In fact, this is itself made up of 3 enzymes. Pyruvate dehydrogenase complex itself is made up of 3 different enzymes. We call them as E1 plus E2 plus E3. That is why it is a complex of 3 different enzymes. So this is the second reaction. Look at this, my friend. We are talking about the second reaction. What is pyruvate? This is pyruvate. And this is converted to what? This is converted to acetyl CoA. What is acetyl CoA? This is CH3, C double bond O, SCOA. This is what is acetyl CoA. This is the acetyl group. Acetyl group. This comes from coenzyme A, CoASH. In this reaction, look at this reaction over here. In this reaction, we also need what is CoASH. This is 3-carbon compound. This is 2-carbon compound. एक carbon कहाँ गायब हो गया? 1 carbon moves out in the form of CO2. At the same time, we also see NAD positive is converted to NADH. Some energy is conserved in the form of reduced coenzyme NADH. NADH is a reduced coenzyme. Look at the reaction. From 3-carbon, we are getting something which is 2-carbon. From pyruvate, we get something which is acetyl CoA. In fact, this reaction is what? This reaction is an example. And who catalyzes the reaction? Reaction catalyzed by PDC, pyruvate dehydrogenase complex, catalyzes the reaction. So this is very important. And we know this reaction में क्या हो रहा है? इस reaction में CO2 भी बाहर आ रही है. इस reaction में NADH का production भी हो रहा है. So this reaction is an example of oxidative decarboxylation. This is oxidative.
सर CO2 के नीचे क्या लिखा है?
PDC लिखा है बेटा, pyruvate dehydrogenase complex. एंजाइम है जो, ये हमारा एंजाइम है, ठीक है? PDC. This is E1 plus E2 plus E3. Remember? ये रहा हमारा pyruvate dehydrogenase complex. This is what I have written here, PDC, which catalyzes the reaction. So, just trying to tell you, second reaction gives us, so पहले हमने 6 कार्बन से स्टार्ट किया, ग्लूकोज से, हमें ग्लूकोज से मिला pyruvate, which is 3 कार्बन. pyruvate माइटोकॉन्ड्रिया के अंदर गया, 3 carbon, and from here we get what is acetyl CoA, which is 2 carbon. Am I clear? So we have seen 2 reactions of respiration. The first is glycolysis, the second reaction catalyzed by PDC, NAD positive converted to NADH. At the same time, CO2 moves out. बात समझ में आ गई? And now we enter the third part of the respiration. By the way, when you talk about respiration, look at the reactions. First is glycolysis, we know this now. The second reaction involves conversion of pyruvate to acetyl CoA. We know this now. It takes place inside the mitochondria. This takes place in the cytosol. Third part involves what? In the third part, ये जो acetyl CoA है, this acetyl CoA is oxidized. This is to be oxidized, मतलब broken down. Large amount of energy is released. Energy would be released when it is oxidized. And that energy is conserved in the form of NADH and FADH. Energy is conserved. In the form of NADH, FADH. We call them as reduced coenzymes. Energy rich. They are energy rich. मतलब कहने का ये, जो हमारी एनर्जी यहाँ ग्लूकोज में थी, एनर्जी विच वाज इन ग्लूकोज, that energy now has been conserved in the form of NADH and FADH. ग्लूकोज की एनर्जी NADH और FADH के अंदर आ गई during the third part. And what do we call these reactions? These set of reactions are, this is the Krebs cycle. This is what is Krebs cycle. Or we call it TCA cycle, citric acid cycle. TCA, tricarboxylic acid. What is TCA? This is tricarboxylic acid cycle. Look at this TCA cycle. Krebs cycle, citric acid cycle, कुछ भी बोल दो. Am I clear? So what is the function of TCA cycle? The function of TCA cycle is nothing else, just to break down acetyl CoA, to oxidize acetyl CoA, which is an energy-rich compound. कोई शक, कोई सवाल पूछो. ये देखो, TCA cycle basically is a set of 8 reactions, roughly a set of 8 reactions. TCA cycle. Location. कहाँ location है? Matrix of mitochondria. All the enzymes are found in the matrix of mitochondria. देख रहे हो यहाँ पे लोकेशन, ये ग्लाइकोलिसिस का कहाँ था? साइटोसॉल, माइटोकॉन्ड्रिया, ये देखो. और थर्ड रिएक्शन कहाँ हो रही है हमारी? माइटोकॉन्ड्रिया के अंदर हो रही है, लेकिन कहाँ पे? इट इज इन दी मैट्रिक्स. TCA cycle. Set of 8 reactions. Roughly, what happens? Starts with acetyl CoA. यहीं से तो स्टार्ट हो रहा है ना सब कुछ. 2 carbon compound, acetate group S में. This combines another compound called as oxaloacetic acid. This is a 4 carbon compound. यहाँ पे condensation हो गया. We have a condensation reaction. 2 carbon, 4 carbon. What do we get? We get a 6 carbon compound. This is roughly what is citrate. सब में रट्टा लगाया है. This is how we get citrate. Citrate converted to isocitrate. Isocitrate converted to alpha ketoglutarate. Alpha ketoglutarate converted to succinyl CoA. Succinyl CoA. Succinyl CoA is converted to succinate. Succinate is converted to fumarate. Fumarate is converted to malate. Malate at the last is converted to oxaloacetic acid. Am I clear? So roughly 1, 2, 3, 4, 5, 6, 7, 8. Finally, again, condensation. Again, we get this. This is what. हाँ जी, बोलो बेटा.
अल्फा कीटो के बाद क्या लिखा हुआ है? At the
Alpha
fourth position.
ketoglutarate. Alpha ketoglutarate. Sometimes we call it 2-oxoglutarate. Another name for this is 2-oxoglutarate, or we can call it alpha ketoglutarate. Okay. Don't worry. I'm just giving you a trailer. अभी तो हम इसको डिटेल में करेंगे. This is what is TCA cycle. Am I clear? And I'm telling you what? That the function of TCA cycle is to break down acetyl CoA. The function of TCA cycle is to completely break down. इसको तोड़ना है हमें, इसको oxidize करना है. We have to oxidize this 2 carbon compound. And when it is broken down, when it is oxidized, a large amount of energy would be released. इसको ऑक्सीडाइज करेंगे, एनर्जी रिलीज होगी. जब एनर्जी रिलीज होगी, इसको वेस्ट तो नहीं करेंगे हम, विल नॉट वेस्ट इट. हम क्या करेंगे, द एनर्जी विच इज रिलीज्ड वुड बी कंजर्व्ड in the form of reduced coenzymes. So let us look at this. Let us look at this reaction. Isocitrate to alpha ketoglutarate. ये जो reaction है, यहाँ पे 1NADH is synthesized. Some energy is conserved over here. Similarly, alpha ketoglutarate to succinyl CoA, NADH is synthesized. Succinate to fumarate, FADH is synthesized. Malate to oxaloacetic acid, NADH is synthesized. Look at this. That means out of these 8 reactions that we see, in 4 reactions, energy conservation will take place. तो 8 में से जो 4 reactions हैं, वहां पे some energy conservation is taking place. And we are getting reduced coenzymes. That means the energy of glucose, we started with the glucose. The energy of glucose now has come inside this NADH, FADH. Am I clear? Yes or no?
Yes, sir.
Yes.
So this is the third. क्या था ये तीसरा part था. Third part was TC cycle. So what is the end product of the TC cycle? Reduced coenzymes. This is it. खत्म और क्या है? Fourth part of mitochondrial respiration. What is that? Everybody knows ETC, electron transport chain. ये हमारा. तो क्या हुआ? Outer membrane, inner membrane. This is the ETC. This is the intermembrane space. We know this. This is the space. This is the outer membrane. ये inner membrane है. This is the inner membrane. ये हमारा matrix है. यहाँ पे TC cycle हो रही थी. This is where the TC cycle is taking place. And what we get? We get NADH. What do we get? We get FADH. What entered the TC cycle? Acetyl CoA. This is the picture. And we know these are energy-rich compounds. This is complex 1, complex 2, complex 3, complex 4, electron transport chain. What is the function of ETC? The function of ETC is to oxidize these reduced coenzymes. इनको तोड़ना है हमें, इनको oxidize करना है. What is the function of ETC? The function of ETC is to oxidize these reduced coenzymes. That is it. ठीक है? क्या हो रहा है? ये देखो. करते हैं चलो इसको oxidize. Let us see this. NADH is oxidized in the complex 1. And when NADH is oxidized in the complex 1, electron is released. When NADH is oxidized, electrons are released. High energy electron has high energy, and this electron will flow through the ETC. Electron will pass through the ETC. Similarly, FADH is oxidized to FAD positive. Again, the electrons would be released. Again, the electrons would be released. ये देखो. Again, they will pass through the ETC. They pass through the ETC from high energy to low energy. When electrons pass through the ETC, they pass from high energy to low energy. That means some energy is dissipated, some energy is lost. So when the electrons pass through the ETC, some energy is being dissipated. And that energy is conserved, that energy is used. The energy which is being dissipated, that energy in fact is used to pump protons. The energy which is dissipated, that energy is used to pump protons in the intermembrane space. Pump protons in the intermembrane space. So Na-DH और Fa-DH की जो energy थी, that energy has been converted into a proton gradient that has been used to pump protons in the intermembrane space. Finally, electron आ गया हमारा. Electron आ गया. And now who will accept this electron? We know this electron finally is accepted by oxygen. Oxygen accept this electron combined with protons, and we get water. We get water. यहाँ पे हमारा oxygen है, यहाँ पे हमारे protons हैं, and we get water. So oxygen accepts the electrons, protons are needed, we get water ultimately. And that is why mitochondria requires oxygen. Mitochondria requires oxygen. Get my point now? So look at this. This is the fourth step. That is electron transport chain. What is the function of ETC? Nothing. The function of ETC is to oxidize the energy-rich reduced coenzymes. When they're oxidized, electrons are released. Electrons flow through electron transport chain down the energy gradient. They flow through the ETC down the energy gradient. Some energy is dissipated, and that energy is used to translocate protons, to transport protons into the intermembrane space.
सर
खत्म.
HCl हाई एनर्जी लो एनर्जी है
क्या चीज?
सर
H मतलब हाई एनर्जी. H मतलब हाई तो होगा ना. H मतलब हाई, Le मतलब लो एनर्जी. Is this clear? So now we see this. So ultimately, what is the end result of electron transport chain? What is the end product of ETC? I can say that the end product of ETC is a proton gradient. When I say gradient मतलब कि intermembrane space में ज्यादा protons हैं और हमारा जो matrix है, matrix में कम protons हैं, less protons. मतलब matrix is alkaline और जो हमारी intermembrane space है, this becomes acidic. So there's a gradient which is created because of protons. More protons, less protons, we have a gradient. So what is the end result of ETC? The end result of ETC, it creates proton gradient, plus we also get water. Am I clear?
सर वहाँ पे आपने एसिडिक लिखे नीचे अल्कलाइन, वो फिर से बता दो.
बेटा इंटरमेम्ब्रेन स्पेस में प्रोटॉन ज्यादा हो गए, तो जहां प्रोटॉन ज्यादा होंगे वो क्या हो जाएगा? एसिडिक. और हमारा जो मैट्रिक्स है, मैट्रिक्स से प्रोटॉन्स चले गए सारे, तो मैट्रिक्स क्या हो जाएगा? एल्कलाइन. Am I clear?
Yes, sir.
So this is the picture. So that means if we start from glucose, अभी energy of glucose कहाँ चलेगी? All the energy of glucose has been converted into a proton gradient. Can I say this? ये gradient जो बन रहा है, the gradient which is being formed over here, we call it proton gradient. Sometimes we can call it electrochemical proton gradient. So we can use different terms to confuse you. I can call it proton gradient. I can call it electrochemical proton gradient. I can call it proton motive force. So one and the same thing. ठीक है? और ये जो protons हैं, these protons which are present in the intermembrane space, they have high potential energy. These protons are found to have high potential energy. आपके घर के ऊपर वो टंकी है ना, the tank of water, the water which is present over there has high potential energy. So similarly, these protons have high potential energy. Okay, so there's a proton motive force which is built up. So this is the end result of the electron transport chain. And finally, we know the energy of glucose has been converted into a proton gradient. खत्म. अब क्या करें gradient का? Let us see the fifth part now. We know after ETC, what do we have? Oxidative phosphorylation. We have oxidative phosphorylation. मतलब क्या? मतलब यहाँ पे oxidative phosphorylation में the ADP is being phosphorylated and we get ATP. phosphorylation हो रहा है भाई यहाँ पे किसका? ADP का. तो ATP बन रहा है और ऑक्सीडेटिव भी है. ऑक्सीडेटिव किस लिए? क्योंकि ये जो फॉस्फोराइलेशन रिएक्शन है, दिस फॉस्फोराइलेशन इज लिंक्ड टू दिस ऑक्सीजन डिमांडिंग ईटीसी. ये जो ईटीसी है, इनफैक्ट दिस ईटीसी नीड्स ऑक्सीजन एंड दैट इज व्हाई वी से कि ऑक्सीडेटिव. एक तो ये मतलब हुआ. दूसरा, बिकॉज द ईटीसी ऑक्सीडाइजेस रिड्यूस कोएंजाइम्स. NADH is being oxidized, FADH is being oxidized, and the oxidation is linked to ATP synthesis. इनका oxidation जो है, that is linked to ATP synthesis. So I can call it oxidative phosphorylation. Something is being oxidized and we are getting phosphorylation done. So we get ATP. So what is the end result of oxidative phosphorylation? The end result of oxidative phosphorylation is the synthesis of ATP. तभी मैंने कहा था ना, 3 methods of ATP synthesis, substrate level phosphorylation. और माइटोकॉन्ड्रिया में जो ATP synthesis हो रहा है, what do we call it? Oxidative phosphorylation. Chloroplast में the ATP which is being synthesized, what do we call it? Photophosphorylation. तो यहाँ पे this is oxidative phosphorylation. The purpose is to synthesize ATP. Look at this. Outer, inner mitochondrial membrane. This is the electron transport chain that we have. And finally, we have the F0F1 particle. This is the ATP synthase. And we have a lot of proton gradient. There are a lot of protons which are present here. There's a proton motive force. The membrane is, I told you, inner membrane is highly impermeable. Impermeable to solutes. That means H ions and they can only pass through F0F1 particle. So H ions can pass through this. And the energy that they have, the potential energy that they have, that potential energy would be used to do certain work. So when these H ions pass through this F0F1 particle, the energy which is present in the protons, that energy is used to produce ATP, to convert ADP to ATP. That is how we get ATP. So we call it oxidative phosphorylation. Am I clear? So we'll talk in detail. Abhi to it looks like very abstract, but when we look at this mechanism in detail, you'll come to know. And we know ये जो F0F1 particle है, we know it can rotate. In fact, this is one of the smallest motors in the world. Smallest motors in the world, it can rotate. And that is how the ATP synthesis will take place. We'll talk about this. There's a model that how ATP synthesis takes place. And the model was given by a scientist called as Paul Boyer. And we call it uncoupling model, that how ATP synthesis takes place by this F0F1 particle. Okay, so we have seen 5 mechanisms. Glycolysis. We have seen conversion of pyruvate to acetyl CoA. We have seen the citric acid cycle or the TCA cycle, Krebs cycle. We have seen the electron transport chain, and finally, oxidative phosphorylation. And this is what is the mitochondrial aspiration. So I hope you have some idea now. You have some broad idea about mitochondria is a very, very big topic. You're clear with the trailer? What are the steps which are involved? Yes or no?
Yes, sir.
Okay, let me
Yes,
confuse you. Let me confuse you. This is the inner mitochondrial membrane. This is the electron transport chain that we're talking about. Complex 1, complex 2, complex 3, complex 4. NADH oxidized to NAD positive. Now the question is complex 1, 2, 3, 4. So the question is, ये जो complex 1, when NADH is oxidized at complex 1, we get electron. Electron is liberated. So do you think this electron would be given to complex 2? Complex 2 will give it to complex 3. Complex 3 will give it to complex 4. And now this will give it to oxygen. And we get water. Do you think it happens this way?
So there are carrier proteins involved.
तो ये सब कैरियर्स ही तो हैं, क्या है ये?
इनके मतलब कॉम्प्लेक्स के बीच में होते हैं.
तो कौन से होते हैं? कहाँ पे कैसे करेंगे इसको बताओ?
सर 2 और 3 के बीच में होता है एक, और 3 और 4 के बीच में साइटोक्रोम होता है.
तो 2 और 3 के बीच में क्या होता है? 1 और 2 के बीच में क्या होता है फिर?
1 और 2 के बीच में नहीं होता.
नहीं होता?
नहीं.
तो मतलब 1 will give to 2 and 2 will give to somebody and that
to
will give to
carrier
3.
protein.
So
But
one will give
if I
to
say,
coenzyme Q and 2 as well.
see, the question is, let us come back. We'll be talking about this later in detail, not to worry. Okay? But what I'm trying to tell you over here, in this case, what we see, कि जो 1 will not give to 2, 2 will not give to 3, and 3 will not give to 4. Always keep this in mind. Okay? 1 will give to somebody which is called as coenzyme Q. So this will give to coenzyme Q. 2, यहाँ पे क्या हो रहा है? FAD, H is oxidized. FAD positive. Again, we're getting electrons over here. So 2 will also give to coenzyme Q. This will also give to coenzyme Q. If you look at it in 3 dimension, if I say this is membrane, अगर मैं ऊपर से देखूं membrane को, this is the upper surface of membrane. तो यहाँ पे मैंने कहा कि complex 1 पड़ा हुआ है, यहाँ पे complex 2 पड़ा हुआ है, यहाँ पे coenzyme Q पड़ा हुआ है, यहाँ पे complex 3 पड़ा हुआ है. ऐसा नहीं है, ये देखो, यहाँ से इसको देगा, यहाँ से भी इसको देगा. You get my point? तो हमने तो यहाँ से section लिया ना, we are looking at a section over here. So 1 can give to coenzyme Q, 2 can give to coenzyme Q. Coenzyme Q is the one which will give to complex 3. This is the one which will give to complex 3. So 1 will not give to 2, 2 will not give to 3. The 1, 2 will give to coenzyme Q, gives to 3, and 3 will not give to 4. 3 will give to somebody who is called as cytochrome C. Cytochrome C, we have a carrier called as cytochrome C. तो यहाँ से यहाँ आएगा. And cytochrome C is the one which will give to complex 4. And this is the one which will now give to oxygen. Plus we have 2 protons and we get a molecule of water. So look at the series, over here. Okay. So small things. अब देखो एक चीज और देखो. Look at this. In this diagram, what do you think is wrong? Except the direction. Direction हमें पता है कि 1 will not give to 2, 2 will not give to 3. But what else do you think is wrong in this diagram? Can you point out? What else is wrong over here?
Proton is not found by every complex.
क्या कह रहे हैं?
Sir, ये second complex
हाँ.
proton
So what we
करेगा
know is, ये गलत है. ये जो है, ये this is wrong. This is entirely wrong over here. Complex 2 will never translocate protons. जो complex 2 है, ये कभी translocate नहीं करता है protons को. Always keep this in mind. This is wrong. 1 can translocate protons, 3 will translocate protons, coenzyme Q can translocate protons, 4 can translocate protons, but never 2. Am I clear? यहां से प्रोटॉन्स जाएंगे, ये भी प्रोटॉन्स दे सकता है, ये भी प्रोटॉन्स को पंप करेगा, और ये भी प्रोटॉन्स को पंप करेगा, लेकिन ये नहीं करेगा. 2 will never translocate protons. This should be clear. ठीक है? So we have seen certain glimpses of respiration today, but there's some problem, again, in my mind, there's certain problem. If you look at the plants, and if you look at the glycolysis in plants, yahan pe bhi ultimately, if you look at the reactions over here, yeah, I'll talk it later, but let me tell you something. Glucose 6 phosphate, fructose 6 phosphate, and this reaction requires ATP. I told you this requires ATP. Okay, there are certain reactions which require ATP. There are certain reactions. Look at the last reaction, PEP to pyruvate. This requires ADP. So in the glycolysis, there are certain reactions which require ATP. There are certain reactions which require ADP. The problem is, if this plant is exposed to nutritional deficiency, अगर यहाँ पे nutritional deficiency है, मैं कहता हूँ कि हमारे soil में phosphate नहीं है, soil has no phosphate, so we have a nutritional deficiency in that case. Phosphate ही नहीं है. हम जो यूरिया देते हैं plants को, जो मोदी जी ने यूरिया के इतने दाम बढ़ा दिए हैं, the cost has gone high. What is urea? It is NPK. जो farmer अपनी खाद डालता है खेत में, that is NPK. We are supplying phosphorus to the soils. So many plants which are growing in the wild, if a plant is growing in the jungle, in the wild, do you think I'll go and supply urea to the plant, which is so costly? That means there's a high chance that the plants which are growing in the wild, they will face nutritional deficiency of phosphate. And in that case, the synthesis of ATP as well as ADP, they would be in short supply. Phosphorus ही नहीं पीछे से. ATP and ADP are in short supply. In that case, glycolysis can be affected. Glycolysis can also stop. When these two molecules are in short supply, due to nutritional deficiency, the glycolysis can come to halt. It can stop. And the plant can even die. There could be this condition. Are you getting my point or not? आपको खाना चाहिए.
क्या बताया soil?
मैंने ये कहा कि सॉइल में अगर फास्फोरस नहीं है, तो प्लांट में ATP और ADP would be in short supply. You get my point? क्योंकि इनके ATP and ADP synthesis requires phosphorus. You have no phosphorus. So they are in short supply in the plants. In that case, the glycolysis can be affected to a limit that the plant can even die. So this is my question to you. Okay. You have to look at the answer for this. We'll discuss this later. So there are more things we have to discuss. Don't worry. We have to start. This is just a trailer today. So if you have some doubts, you can ask me. Regarding today's topic, I'm only talking about this lecture over here. Not doubts outside the lecture, but doubts within the lecture. We have yet to start. This is just a, because mitochondria is a big topic. You have to have that a broad idea how we relate things. So all the energy in the ATP now, is coming from the proton gradient, and the energy to create this proton gradient ultimately comes from glucose only. Everything is being pumped by glucose. The greasing is taking place by glucose. see something. Roughly. This is what is happening in mitochondria. This is mitochondrial ETC. Is this clear? We know this is mitochondrial ETC. Look at this now. Look at the photosynthetic ETC. Chloroplast also has electron transport chain. Chloroplast also has ETC. Look at this. You remember? If I draw the structure of chloroplast, ये अगर chloroplast है, यहाँ पे भी double membranes and you have grana interconnected, stromal lamellae, granule lamellae, all this is there. If I take this membrane from here, I'm taking this membrane to the other page. I take this membrane over here. किसकी membrane है? This is the membrane of the grana. This is a granule membrane, thylakoid membrane. Or I can call it thylakoid membrane. यहाँ पे भी ETC है. Everybody know. Photosystem 2. Photosystem 1. Who connects the two? Cytochrome B6F. Complex. याद है ना? Cytochrome B6. This is what we have. यहाँ पे क्या है? Plastoquinone. यहाँ पे क्या है? Plastocyanin. How we start? Water. क्या हो रहा है? पानी टूटा. Protons. Plus we also get oxygen. Oxygen मिली, protons मिले. Plus we get electrons. Electrons pass through the ETC. Look at this. Electrons will pass through ETC. And finally, electrons come over here. What is happening here? NADP positive is converted to NADPH. And this is the end. What is happening in the photosynthesis? In the photosynthesis, what you have to look at, in the photosynthesis, we start with water and we end with NADPH. We start with water and we end with reduced coenzyme. What about mitochondria? We start with what? NADPH. And we end with what? Water. मतलब photosynthesis का opposite है. Respiration में something happening which is opposite of photosynthesis. Photosynthesis में we start with water and we end with reduced coenzyme. And respiration में we start with reduced coenzyme, end with water. Second thing, एक और चीज देखो. Respiration में oxygen is needed. We need oxygen. Photosynthesis में oxygen is liberated. Look at these changes. देख रहे हो? मतलब the two mechanisms, photosynthesis and respiration, which are very important for the entire life to exist, they are nearly opposite in nature. Are you getting my point?
Yes, sir.
So if you have some doubts, you can ask me now.